What makes you look different from your siblings? What makes you look alike? In this topic of genetics, we're going to be defining and looking at some of the terms of genotypes, phenotypes, what is dominant versus recessive, and looking at those characteristics. Humans have long been interested in heredity. We look at crops and animals to give them traits that are more useful, for example, certain breeds of dogs, or even plumper tomatoes. The patterns that Mendel studied forms this topic of genetics. The study of heredity began with Gregor Mendel, who was a monk. He went to Vienna to study math and science, and he was interested in getting to know the new colors of flowers. He always asked the question, why? Why were they different? Why did some look the same? And he studied the pea plant. Mendel studied the pea plant because it was a good subject for studying heredity for the following reasons. There were several traits that showed different forms. For example, color, pea color, or the seed color, the pod color, even pod shape, even flower color. He used this for self-fertilization and cross-fertilization, and they found out that it was possible. It was easy to cultivate, and the plants produced many offspring. What Mendel did is he first worked with true breeding plants. What a true plant is that over generation after generation, that plant produced flowers that were purple. Or after generation, the plants produced white flower. What he did is he took a transferred pollen from the stamen of a white flower. So you can see here, remember the stamen of a white flower, so that's the male part. What he did is he transferred that pollen to the carpel, or the female part of the flower. What happened next was that fertilization took place, and they produced seeds, which then grew and matured into a pea pod. The result was all purple flowers. The question is, why? On this slide, you can see some of the characteristics that Mendel was able to study. On the bottom, you can see we have purple flowers. And some of the other characteristics were whether or not the seeds were wrinkled versus smooth, green or yellow. Here is the pea pod, whether it was inflated like a balloon or constricted. Right here, yellow or green. Another characteristic that he looked at was the seed shape, whether it was round versus wrinkled, as you can see on the other slide. What we're going to do next in the next two slides is show you a step-by-step process, and then we'll go back to the flower color. The first step is that you need to establish true breeding characteristics, which means, again, all offspring time after time would only display one type of trait. A good example was all offspring with purple flowers. There is no variety, only purple. What Mendel then did is he crossed the true breeding purple and he crossed it with a true breeding white flower. This represents the parent generation, or P cross. The offspring of those parents will be the first generation, and that is called the F1. An example is your parents represent the P cross. You are the first generation of your parents, so you are the F1 generation. Here again is the purple flower example. We took a true breeding purple with a true breeding white. That means all the flowers in this generation were purple, and all the flowers in continuous generations were white. They crossbred them, which means they reproduced, but they all produced purple flowers. Why was that? We still don't know. They then took two of these purple flowers and crossed them again. So two F1s, F1 times an F1. They got 3 purple and 1 white. The white showed up again. Why is this? Let's recap. We took a true breeding parent, so this is the P generation, and these is a P generation, and they crossed it, and they got an F1. All were purple. This means that purple is dominant over white. White is recessive. It's not strong enough. After that, they took two F1s, so F1 times an F1, and they again got 3 purple and 1 white. The white again reappeared. We still don't know why. That should be in our next presentation. We want to know why that white one reappeared. Well, when we have two F1s in that generation, when we cross them, the missing trait, that white flower, reappears in some of the plants in the F2 generation. This is a ratio, and it will be in the simplest form, and we're going to work this out in some problems. In this topic, we're going to be describing how gene expression is regulated through the process of meiosis, something that you should be familiar with already, as well as identify and explain the laws of segregation and the laws of independent assortment. Mendel's hypothesis was based on the results of the experiments using the pea pods. This makes up the foundation of genetics. We're going to be looking at the copies of a genes or the different versions. One of those is dominant. It's the expressed form. It's the strong one. It's used with a capital letter. And then one is recessive. It is the one being shadowed. The trait is not expressed. It is expressed in a lowercase letter. These alleles separate independent from one another. Recall in meiosis, where we have two haploid cells. This is haploid, the egg, and the sperm is haploid to make a diploid cell. You can see here that we have a capital letter that's going to be the dominant, and then we have the lowercase, the recessive. Together you have a capital and a lowercase. This is called heterozygous. To look a little closer, right here is a homolog, or a chromatid. Right here is another homolog, or a chromatid. Together, they're going to make a chromosome. The locus for that flower gene color is a location or a band on the chromosome. As you can see down here, we have a lighter pink and a darker pink. Some terminology to know, for example, would be seed color. Dominant takes the capital form. So in this case, the seed color, yellow is dominant, so the letter is going to be the capital Y. The green, though, is a lowercase y. At this point, you do not have different letters. We're only looking at seed color. One trait, one letter. Homozygous would be two alleles that are the same, which means dominant yellow or recessive green, two of the same. Heterozygous is different. One big, one little. It's still yellow. Why? Because the larger Y is dominant. It overshadows the baby Y. The genotype is the genetic makeup, which means the letters. That's the biggest thing to remember. All students usually forget that, or they mix it up. The letters are the genotype. What it looks like, what color is it? Is it yellow or green? That is the phenotype. So phenotype is the physical characteristics. Genotype, letters. Can you recall the F1 cross? Here we had 3 purple to 1 yellow. That is a phenotype ratio of 3 to 1. But now let's look at the genotype. We have 1 after doing a Punnett square, which is homozygous dominant. We have 2 heterozygous, which is a big P and a little p. And then we have a homozygous recessive. In this case, the ratio will be 1 to 2 to 1, or the genotype ratio. Again, the phenotype ratio for an F1 cross, which is what we just did, will always be a 3 to 1 ratio in the phenotype. Going back to your knowledge of meiosis, law of segregation states that 2 alleles for a trait will separate when gametes are formed. So in that anaphase, remember, they are going to separate, and it's independent of one another. So you don't know what trait you're going to get. Remember, in the end of meiosis, you have 4 different daughter cells that all look different. Let's look at this cross one more time. Here we started off with the P1 generation. We have a true bred purple and a true bred white. Here we have their parent generation. It resulted in an F1 generation of all purple flowers, but they were all heterozygous, and we can see this in a Punnett square. Okay, and we'll fill that in on the board. Here we took two F1 generations and we crossed them. The results are a little bit different. We have now a phenotypic ratio of 3 to 1. 3 purple to 1 white. All of a different genotype. We have one that is homozygous dominant. We have two that are heterozygous. And then we have one that is homozygous recessive. To recap, the law of independent assortment. These alleles of different genes, they will also separate independently from one another during gamete formation. Example, genes for color are going to separate independently of genes for height. Just because a seed is yellow does not mean it's going to be a shorter seed or a taller plant.